So our next speaker is Fred Rombouts from Augustine Therapeutics, and he's going to talk to us about HDAC6 inhibitors.
Thank you. First of all, thank you for inviting me and talk about our programs. Let me see if I can figure out the remotes. Yeah. Okay. So this slide is something you're all probably familiar with. As you know, CMT has several subforms, with CMT1 being the most prevalent form, and CMT2 the second most prevalent form, and then a number of less prevalent subtypes as well. We have looked mostly at CMT1, but also a little at CMT2. CMT1 is characterized, as you know, by demyelination followed by axonal degeneration, while CMT2 is characterized primarily by axonal degeneration, leading then to the phenotypes you see there. Now, axonopathy as such is characterized by defective alpha-tubulin acetylation, microtubule instability, and the blockade of axonal transport. In comes HDAC6, which is, for all clarity, not an epigenetic histone deacetylase, but a cytosolic deacetylase. And that's important to realize because its function is really in the cytosol. The substrates are in the cytosol. It's a class 2B HDAC, which is unique in that it has two catalytic domains, a CD1, CD2, but also a zinc finger ubiquitin binding domain. An important target of HDAC6 is alpha-tubulin. There's also HSP90, cortakin, and a few others. But so it's important to realize that it's not involved in epigenetic regulation, but rather in for instance, the acetylation of alpha-tubulin. The way to modulate HDAC6 is also important in treating CMT. So it's important that you inhibit the catalytic domain, the CD2 domain, whilst not affecting the other functions of HDAC6, which makes a inhibition with a small molecule, for instance, a preferred option over, for instance, degrading HDAC6 to allow the beneficial effects through protein-protein interactions still to happen. Once you inhibit HDAC6, promote alpha-tubulin acetylation, you will allow the stabilization of microtubules and the transports of cargo along the microtubules. What's also important, and for a drug discoverer, is to know that the knockout mice are viable, they have the same lifespan as healthy mice, and there's no toxicity reported. This is a picture showing more in detail what happens when you inhibit HDAC6. So on the right-hand side, you see the disease state where the axonal transport is impaired. Once you block HDAC6, you will hyperacetylate, so it's like paving the road again so that the trucks can pass to transport the cargo along the axons, including mitochondria, and this leads to improved axonal health and regrowth of the neurons. Now, the target has been around for a long time, actually, and the major challenge in developing HDAC6 inhibitors has been the chemistry, finding safe drugs that you can dose to patients chronically. People have found, for instance, hydroxamic acids, oxadiazoles, but they have been associated with toxicity, with off-target activity, and hence have struggled to move through clinical trials. So what we have done is we've started off with a high-throughput screening of 85,000 compounds, a commercial library. We have excluded all chemotypes that look like hydroxamic acids and the fluorometal oxadiazoles. And we have identified a novel chemotype that we have characterized through our internal cascade, which is in-house developed biochemical assay, but also a nanobread, so an in-cell target engagement assay, and then a imaging assay, so through immunocytochemistry, where we can image both the acetylation of alpha-tubulin versus the acetylation of histones, showing then whether the effect is purely on alpha-tubulin or also on the epigenetic HDACs. And through that funnel, we have identified then molecules that are both unique from a chemical point of view and highly selective for HDAC6. That series has been optimized and led to the discovery of our lead molecule, HED216, of which I will show some exciting data. But in the meantime, we've also developed follow-up candidates which are peripherally restricted, such as HED216, but we also managed to get brain-penetrant leads, which are now in lead optimization stage. So these are some data on our lead molecule, HED216. So with our in-house developed biochemical assay, we demonstrated that this compound has 90 picomolar activity for HDAC6, so this is probably the most potent HDAC6 inhibitor around. Previously, this assay also was optimized for HDAC1 and 3, where we show nice selectivity versus these epigenetic HDACs. Previously, at Reaction Biology Corporation, we also check the selectivity for a broader range of HDACs, showing that it is selective for HDAC6. We've demonstrated also that it binds to the CD2 domain of HDAC6, so we see a temperature shift here, stabilizing that CD2 domain in the same range as well-known hydroxamic acid HDAC inhibitors such as trichostatin A and duvastatin A. In the nanobread binding assay, we showed double-digit nanomolar potency for HDAC6 CD2 domain, the human one, with selectivity over the closest related, so also a 2B HDAC10. And then in the functional assay, looking at tubulin acetylation versus histone acetylation, we showed double-digit nanomolar potency for inhibiting the acetylation of alpha-tubulin with a large window of about 100-fold versus the acetylation of histone H3. So with those data, we progressed the compound through the, let's say, regular DMPK and tox profiling, and eventually to an efficacy model, which the first study is shown, of which the first study is shown here. So what we did here is to dose HED216 in CMT1A C3 mice, so they have multiple copies of the human PMP22. We dosed the compound for 4 weeks in a preventive setting, so we started dosing at 4 weeks, where the mice do not show yet the phenotype, and continued dosing until they were 8 weeks old. And what you see in the red bar is that the untreated mice clearly develop deficits in the compound muscle action potential, in the grip strength, and in the ball withdrawal, so motor function related and axonal regeneration related outcomes, while the treated mice with 30 milligrams per kilogram of HED216 show a prevention of that, or partial prevention of that phenotype occurring. So they basically partially rescue that deficit both in the CMAP as in the motor function. And this was associated with an increase of the acetylated tubulin in plasma. Now, for all clarity, there is no alpha-tubulin in plasma, so this is from ruptured blood cells in the preparation of the plasma, and we optimized that assay in the meantime to have a stronger signal to noise. And we also saw a dose-dependent decrease of neurofilament light chain in those treated mice. So with those encouraging data, we also progressed the compound to a a curative setting. So here we dosed 8-week-old CMT1A-C3 mice for 1 month with HDAC6. So here they already developed the deficits, and what you see is that at 12 weeks the untreated group progresses, while the treated group shows a dose-dependent reversal of the phenotype. With HDAC6, we tested 3 milligrams per kilogram BID up to 30 milligrams per kilogram BID, and the efficacy starts showing up already at 10 milligram per kilogram BID. It maximizes at 30 milligram QD per kilogram QD, while it rather plateaus than if you dose higher. So it seems that we are in the efficacious range with these doses. Again, we show here effects on the CMAP, but also nerve conduction velocity and on motor function. So in all three, we see significant reversal, rescue of the symptoms. This again was associated with a dose-dependent increase of acetylated alpha-tubulin, so that's encouraging because that probably gives us a good biomarker to see target engagement in the clinic. And it also was associated with a dose-dependent decrease of neurofilament light chain, as you know, a marker for neuronal damage. When you look at histology, this is the wild type mouse sciatic nerve. This is how it looks in a CMT1A mouse once it has developed the phenotype. And with HDAC6, you can see that we are able to reverse that phenotype, rescue it completely with a lot of axons that are myelinated. We also have, in collaboration then with Professor Ludo van den Bos, who is the scientific founder of our company, looked at human iPSC-derived motor neurons from a CMT2A patient having this mutation. And in those neurons, we have demonstrated that HDAC6 rescues the acetylated alpha-tubulin to a similar extent as tuvastatin A. Again, a well-known hydroxamic acid, HDAC6 selective inhibitor. And this confirmed target engagement is then expected to translate into robust efficacy in a mitofusing 2 mouse model, as was demonstrated by 2 separate groups previously. So with that, I hope I've shown you that HDAC6 is a promising new candidate for treating CMT1A. So it's a potent, selective, orally available and peripherally restricted non-hydroxamic acid and non-oxidizole HDAC6 inhibitor, which makes us believe that it's a best-in-class molecule. We have a use patent application granted for the use of HDAC6 inhibitors in CMT subtypes in major markets. I hope I've convinced you that it shows robust efficacy in a symptomatic CMT1A mouse model. It reverses and prevents the disease phenotype. It rescues acetylated alpha-tubulin in plasma and in sciatic nerves. And it improves the demyelination and reverses the axonal damage biomarker neurofilament light chain. It also rescues acetylated alpha-tubulin in CMT2A human iPSC-derived motor neurons. What I haven't talked about, but is of course very important for its progression, is that it showed clean in vitro safety and toxicology profile compared to other HDAC6 inhibitor classes. It was well tolerated both in the non-GLP as in GLP tox studies, and we achieved a high safety margin. So now we're close to completing the IND-enabling studies and plan to start a phase 1A study in 2025. So next year, moving to a small patient group and a larger 2A study in 2026 with CMT patients. Lastly, I'd like to thank the team. So we are located in the bioincubator in Leuven in Belgium. The team is Sylvain Selanier, our CEO, who was here, I believe, last year. Jonathan van Eyl is our head of pharmacology. Cedric is the head of early drug development. Luc is our CMC consultant, and Lise is the technician who runs the assays in our labs. We also, of course, thank Ludo van den Bos, our scientific founder, but also advisor, Rob Pryor here today, and Wendy, who also did some of the in vitro work. And then, last but not least, we thank our sponsors, so Flyo, a Flemish fund, and of course the CMT-RF, who has supported us very well, and also supported the in vivo study I've shown, and the other investors that have supported us so far. And with that, I thank you for your attention, and I'm open for questions.
Okay, we have time for a few questions. Brett?
Thanks for that great talk. I'm curious what you think about the mechanism of action of HDAC6 inhibition, because there does seem to be some data that suggests there's a dissociation between the effect on tubulin acetylation and the therapeutic effect.
That's a fair point. So acetylated alpha-tubulin is not the only substrate of HDAC6, and we seem to affect myelination, which can't be related to tubulin acetylation alone. And you've probably seen the paper from the Korean group where they tested CKD504, so one of the former leads of CKD Pharma, where they've shown that the effect seems to be related to more the chaperones or the chaperone pathway being activated. So their hypothesis is, and that's the one we currently also believe, is that through the acetylation of HSP90, you can also upregulate HSP70, which then starts folding and refolding the aggregated PMP22, which in turn helps either it getting degraded then through the proteasome, or maybe also triggers other pathways. So there's multiple roles of HDAC6, many of which are not fully understood, but that is our current thinking in how it works in CMT1A.